Today, our presenter is Dr. Roger Lewis, who is a senior medical scientist at Berry Consultants and professor of emergency medicine at the David Geffen School of Medicine at UCLA. Roger is also a senior statistical editor for JAMA and was a faculty member of the CTMC for many, many years. So we are so grateful to Roger for presenting this session today, which is titled Tale of Two Documents, Grant Applications and Clinical Trial Protocols. Thanks, Roger.
Great. Thank you. It's a pleasure to be here. It's good to see some familiar faces. So these are my financial disclosures, which have already been mostly mentioned. I'd like to acknowledge support from National Institutes of Health and NINDS for a number of activities. And as mentioned, I also work as an editor at JAMA. So I'd like to start by sort of setting the stage of the challenge or regarding the challenge of the documents you have to manage when you're putting together a grant application for a clinical trial and with the associated clinical trial protocol. So you have the grant application itself. So that's the document submitted to and reviewed by the funding agency with the hope that you get funding. There's a proposal. The difference between a grant application and a proposal is that a grant application is just what it says for a grant, whereas a proposal is a similar document submitted and reviewed by a source of financial support, but you're seeking a contract to perform proposed work. The difference is that a contract is usually more specific and has more restrictions regarding what you can do with the money. A clinical trial protocol is a document that details the design, conduct, and planned analysis of the clinical trial. It is intended to ensure that the trial could be run by people who weren't originally involved in designing it. The statistical analysis plan document is a much more comprehensive description of the statistical design, conduct, and final analysis of a clinical trial. So it will repeat some of the information in the clinical trial protocol, but in much more detail. A manual of procedures is a document that may or may not exist for a particular trial that you're involved with or trying to lead, but it's a detailed and often frequently updated description of detailed procedures. So how bloods are drawn, how samples are handled, how they're shipped back to a central lab, who to call in case the web randomization system doesn't work, all those sorts of details. One of the main differences between a clinical trial protocol and a manual of procedures is that the manual of procedures can be updated frequently without it being an amendment to the protocol that requires re-review, for example, by an IRB or approval by your funding agency. A DSMB charter is a description of the function, structure, function, and responsibilities of a data and safety monitoring board, sometimes also called a data monitoring committee. And it's something that's important to have in place before the first meeting of such a committee. We're going to focus on these two documents and what are the differences between them, because one of the common pitfalls for earlier stage investigators is confusion about what goes where or making these documents too similar without acknowledging that their purposes are really quite different. So when thinking about a grant application and clinical trial protocol, both are required, neither are sufficient. And I'll come back to the end and mention about sort of what happens if you try to get away with only using your grant application and the description of your trial within that as your protocol. So as I mentioned, the purpose of the grant application is to get funding, whereas the clinical trial protocol is to precisely define how you could conduct the clinical trial. It's much more operational. The target audiences are different. The target audience for the grant application are those who decide whether to give you the money to do the study, whereas the target audience for the clinical protocol are the people who either have to approve the study or who are actually going to be conducting it with you, your investigators, co-investigators, etc. The content of the grant application, which we'll go through in some detail, has background, rationale, specific aims, research plan, team qualifications, and budget. Whereas in the clinical trial protocol, there's going to be very brief background information, some overall design objectives, but then there's going to be enrollment and consent procedures, data collection procedures, how adverse events are reported, analysis plans. The grant application, as I'm sure many of you know, has very rigid structure regarding the sections and the limits, whereas clinical protocols do generally have similar structures to each other, but the lengths can vary tremendously from a few tens of pages up to hundreds of pages. So this is a comment I found that was attributed to Peter Drain, who was at University of Pittsburgh at the time. Says the grants writing is getting the money. Science is what you do when you get the money. They are two distinct activities. First, get the money. Second, decide on the best science to do with that money. Getting those, basically saying that those are not the same thing. So here are some example outlines, or an example outline, excuse me, of a grant application. So you have your introduction, specific aims, research strategy. This is pulled directly from the structure of an NIH grant application. There's some things about the progress report or publication list to show what work you've already been able to accomplish, how you're going to structure the leadership plan especially if there's multiple uh, PIs, contractual arrangements, letters of support. Things like the introduction and the letters of support are there to point out how important it is, the work that you're doing, and that others think it is important as well. In contrast, a clinical trial protocol will typically have it outline something like this. There'd be a title page, background and rationale, study objectives, hypotheses, study design, etc. And it's very tempting to try to make equivalent or act as if some of these are equivalent, like the background and rationale. I'm sorry, that the study objectives, number 3 for the clinical trial protocol, is the same as the specific aims in a grant application, whereas that really isn't the truth. So first, I want to emphasize the relative length and detail of these different sections in grants and protocols. Because one of the things that um, sometimes gives a hint that someone is very early in their research experience is there's too much detail in a document that doesn't need it and not enough detail in a document that does need it. So, for example, for the scientific background, which is explicitly stated in the grant, it's contained within the introduction and a protocol. The grant needs to be very detailed because its purpose is to convince the grant reviewers and grant review agency that the background information that exists is motivating of the research, that it suggests this research is needed, it's the next step, it fills an important gap in knowledge or our ability to care for patients. Whereas in the protocol, the assumption is that the funds to run the study exist, it's already been deemed important. The only scientific background there is the background necessary to understand the terminology or purpose or overall purpose. You're no longer trying to convince somebody that the study should be done. For specific aims, we'll talk a bit about it. There's a set of specific aims incredibly important in a grant. And in fact, the structure of the data collection, data analysis needs to be tightly tied to the structure of the specific aims. A protocol has something called study objectives, which are often much more operational, but they will include some specific statements about what hypotheses the clinical trial is intended to demonstrate or disprove. The overall research strategy will be very detailed in a grant. It's actually less detailed in the protocol because the overall research strategy is the overall approach to what you're doing. You're going to do some laboratory work, you have an animal model, then you're going to do a clinical trial, etc. Whereas in the protocol, the detail goes into the detailed study procedures. How exactly are patients going to be identified, consented? How are you going to handle people who drop out prematurely? How do you do a follow-up, a closeout visit? Those sorts of things. How are data collected? That's where all the detail is in the protocol. The information in the grant, in contrast for the study procedures and data collection procedures, is just detailed enough to know that you actually have a plan, a concrete plan. But this is probably a good time to point out this is why it is so useful if the grant mechanism allows for you to make a protocol available to reviewers who want more detail in these areas. You can't put the detail in the grant because you'll run out of the pages necessary to actually explain why the research is important. But giving the reviewer who's interested or has a question a resource that they can then refer to is a wonderful thing to do if the grant mechanism allows it. Some do, some don't. The statistical analysis plan for your final data analysis is moderately detailed in both a grant and a protocol, but it is very detailed in the SAP. So the SAP will have much more detail, and the SAP, the statistical analysis plan, will override both the grant and the protocol if there are discrepancies. So if the grant and protocol fail to mention something, but it's included in the SAP, in terms of the analysis, the SAP takes precedence. If you have an interim analysis plan in your study, so you're running a study that's long enough and has either outcomes or safety information accrue quickly enough for individual patients so that it makes sense to do an interim analysis, the grant will mention it. And if it's pretty straightforward, it may be just a few sentences. If it's more detailed, there may be more information. But the statistical analysis plan has to lay out in quite a bit of detail. The protocol will only mention that it exists. There will be virtually no statistical detail in the protocol itself. Adverse event reporting, it's important to mention in the grant that you'll be doing this and that it'll be monitored, but the details are in the protocol. When you're submitting a grant, it is a wonderful thing if you have a detailed statistical analysis plan and you can submit it with the grant and it can be reviewed. Again, it depends on the mechanics and the restrictions on the grant mechanism. When you have it, it shows that you've thought about not only why you're doing the study, that's in the grant, how you're going to do the clinical aspects of the study, that's in the protocol, but the details of the analysis procedure. So if you can find a way to get that together and make sure it's a good version of it, then it's very good to submit. In a protocol, it's absolutely necessary. Now, you want the statistical analysis document to almost always be a separate document or an appendix to the protocol so that it can be revised without amending the entire protocol. If every time you have to change some detail in the statistical analysis plan, you have to make that an amendment to your protocol and send it back to the IRB, that can be a nightmare, and it's a self-inflicted injury. You don't have to do that. The DSMB charters and the manual of procedures are very rarely included in grant applications, but they are separate documents that are necessary to be included with the protocol. I should be more careful here. The DSMB charter is a necessary document if you have a DSMB. A manual of procedures, if you have it, needs to be included or mentioned in the protocol. One of the pieces of art in managing all these different documents is being very careful to avoid duplication between the various parts of the protocol. So, for example, you can have a very broad overview of the statistical analysis and the protocol, and then explicitly say that the details are in a separate statistical analysis plan. So you've incorporated it by reference. When you do that, it means every time you change your SAP, you don't even need to look at your protocol. You know, there's nothing that you're going to need to change. So, as I mentioned before, a common area of some confusion is the difference between specific aims and objectives. So, the specific aims of a grant are a relatively broad set of separately and precisely defined and achievable goals with stated metrics for success for each of those specific aims. I want to make a point or emphasize the difference between specific aims that are interconnected versus interdependent. So interconnected means that the specific aims together create a related set of scientific goals, all of which, if achieved, will have completed you the picture or completed the answer to the problem that you're trying to solve or filled the gap. You'll know whether there's target engagement of the molecule. You'll know whether there's a pharmacodynamic effect, meaning, does it actually change the physiologic process? And you will know whether or not there's a change in clinical outcome, meaning a way a patient feels, functions, or survives. Interdependent means that one of the specific aims, one or more of the specific aims, is only relevant if you are successful on another. They are contingent upon each other. I cannot tell you how often you will see a criticism of specific aims in a grant that says, if specific one fails, it is unclear what the investigator is going to do because specific aims 2, 3, 4, 5 all seem to assume that one will be successful. If specific aim one is to demonstrate that a drug is safe before you deploy it in a larger study. If it's not safe, what are you going to do? Probably not a great example because in drug development that actually does kill the program. But you can picture ones in which it's even a laboratory-based study in which interdependency makes it very unclear that there is a long-term plan for the research program that doesn't make unproven assumptions about the success of specific aims still needing to be addressed. The specific aims are often focused on demonstrating scientific principles like target engagement or pharmacodynamic. Pharmacokinetic is things like drug levels over time. Pharmacodynamic is the effect of the drug on the physiologic process it's intended to influence over time. So insulin pharmacokinetics is how much is insulin around. Pharmacodynamics is how much hypoglycemic effect there is of insulin. Those are important terms to keep distinct in your mind if you're working with drugs. The objectives of the trial should align with the specific aims, and some of them may actually be identical. You can have a specific aim that says to demonstrate, drug X reduces symptom Y in population Z. That's fine in both places. But in contrast, in general, the clinical trial objectives are a small set of goals that are narrow in focus and directly tied to the primary, secondary, and sometimes safety endpoints of a clinical trial. So if your primary endpoint is the modified Rankin scale at 90 days, then your primary objective is going to be to demonstrate an improvement in the distribution of the modified Rankin score at 90 days with treatment X. You want to avoid your objectives being things that are process goals. Setting up sites, enrolling a given number of patients are not objectives of a trial. They may be milestones for your grant, but they are not objectives for the trial. And so I gave an example of how specifically worded a clinical trial objective should generally be. I guess I was supposed to come to that point last. Okay. So I even looked up on the internet, interconnected, interdependent, to sort of prove to you that this is really a distinction. And the internet agreed with me. Since you don't know the order of me looking and writing the slides, you don't know whether that's surprising or not. Okay. Now let's talk about how grants are reviewed. So there are three main factors or domains of grant evaluation, and then there are things called additional review criteria. So factor one is the importance of the research, significance or innovation. The reviewers are going to learn about that in the introductory and background information regarding the significance of the problem. In your research plan, you're going to call out areas of innovation where you plan to do something differently that you think solves a problem that has hampered the field and not previously been solved. Factor two in rigor and feasibility. Um, this is um, these are really two dimensions. So rigor is the things that you do to ensure that the answer you get is the right answer. So things like blinding a treatment assignment or making sure people who assess the outcomes of patients with neurologic disease are blinded to the treatment assignment, even if the patient is not. So blinded outcome assessment. Ensuring that there aren't incentives for patients to misreport their symptoms either in one direction or the other, sources of bias. Feasibility is your ability to actually do the trial as intended and with good fidelity to the trial protocol. So that's a different characteristic. But the reviewers at this stage are asking themselves, if you were able to complete the trial as designed, do I believe there's a very high probability the answer you get, positive or negative, is going to be correct and without substantial bias? Is it going to be reproducible? Could others do the same study again and likely get the same or similar result? Feasibility is the question of, do we think you can actually do it? Do you have enough experience? Are the personnel um, adequately experienced or equipped? Do you have a number of sites that can actually enroll the number of patients you need? Are you actually going to be able to get the patients to come back at 30, 90, and 180 days, whatever it is? Factor three is the expertise and resources. These are sort of foundational to the entire program. Does the investigator team have the depth, breadth, and diversity of both expertise, so knowledge and experience where they've done similar activities before to suggest that they can pull off what has been written down? And is the environment supportive? I think it's goes without saying that there's a lot of concerns about research environments in lots of different contexts, and we want to know that if we award you the money, that your activities will not be crippled by an environment that can't support that. One of the things that is incredibly important in environments is the protected time of investigators, especially early stage investigators, and whether the environment really guarantees you the time to do the work that you're planning to do. I would say more and more when we read letters of support from department chairs, they are less clear than they used to be on protected time and protection from clinical responsibilities. And that's something that at least I pay a lot of attention to when I'm reviewing grants. Additional review criteria can include things like data sharing agreements, human subjects protection, um, and then there are things like review of the budget that don't affect your priority score directly, but it may warrant comment and require revision of your plan if the funding agency decides to move forward. The rigor and feasibility part is really informed by material in the clinical trial protocol. This is where we look to see whether or not you've thought about the procedures in enough detail so that we believe you can do it in a way that's reproducible. So I'll just give a concrete example. So say you plan to do a 6-minute walk. People who do 6-minute walk tests in either the setting of cardiopulmonary or neurologic disease know that it's a test that is highly dependent on the details of how it's administered. So if you simply write in your clinical trial protocol, we'll do a 6-minute walk at day 30, that doesn't tell me that you have any idea how complicated that is. If instead you say we will do a 6-minute walk in at day 30, it will always be done at this location We will use trained administrators who understand coaching limitations. They'll have a set of guidances regarding how they are to coach or encourage patients. It will always be level, the lighting will be, et cetera, et cetera. We'll make sure that the patient has not exerted themselves to get to the testing facility. All those different things, and then reference um, um, one of the many publications about issues in the administration of the 6-minute walk. Then it's shown you've thought about it. This isn't just something you thought, oh, this is a good data point I should have. I've seen it in the paper, but I really have thought about the details about how I'm going to do this. There are lots and lots of outcome procedures for which spending time reading about the problems with them so that you can address them in your protocol to make sure they're assessed properly goes a long way to convincing a reviewer of the grant that you really do know what you're doing. This is why, if you can at all include the protocol, it's so helpful. You couldn't possibly include this detail in the grant application. I will make the point that there are some tricks to making clinical trial protocols available. So, for example, if there's a limitation on appendices or supporting documents in your grant application mechanism, so you cannot include a protocol, you can set up a public website for your trial. It's also useful for other things like getting community engagement or public input into your trial design. And you can post the protocol there and you can put the URL in your grant. The grant reviewers have absolutely no obligation to look at any materials outside of the grant itself. However, if you, in your grant application, say additional details are available in the protocol posted publicly here, then if someone has a concern and is going to write a critique of, you say, a data collection procedure, my experience is that most reviewers will go look at that and they'll actually appreciate the fact that you've given them additional information so that they can make sure that their critique is on target. So, number of bottom lines here. First, very little of the text. I mean, I originally wrote this slide saying none. In either document can be repurposed directly for use in the other. So if you're copying and pasting, that's a bad situation to be in. It's it's attempting to do so, but if I'm sorry, it is tempting, but attempting to do so is going to be completely obvious to anybody who looks at both documents. It's going to seem like you didn't have enough time to think about these carefully, and therefore we worry that you have had enough time to think about the whole project. And it's likely to result in both the inclusion of unnecessary information, like too much detail in the grant on the study collection procedures, and the omission of important information um, in others. So if you try to create a study protocol by copying your grant text over, you'll end up with a protocol that has way too many holes in it. Bottom line 1.1, there are sections in a grant application that really have no substantial equivalent or corresponding section in the clinical trial protocol. So don't try to move any of this into the protocol just because it was in your grant. It just doesn't go there. And then there are many sections in the clinical trial protocol whose content is really only briefly summarized in the grant. It's just mentioned. Adverse events will be collected both by spontaneous report and by structured interview on days 30 and 60. And then in your protocol, you have exactly what that structured interview looks like. You may even have a script for the telephone for a telephone survey of people for adverse effects, et cetera. That detail is just in the protocol. So bottom line number 2. So whether you draft your grant application or protocol first is a matter of personal preference. And I would say if it's like any other previous year, you'll find that even within your instructors in the course, there are going to be people who feel one way or another. I feel obligated to point out the advantages of writing the protocol first. Okay. And I realize this is not the most common approach. So the reason for writing the protocol first is the general point that it is much easier to simplify detailed information to create a more terse, grant-appropriate description of the clinical trial than it is to take a brief description that was in a grant and expand it into a clinical protocol. It's just much easier to trim down than to add. The second point has to do with the thought process that goes on, or the cognitive process as you work on a detailed protocol. As you're writing a detailed protocol, you will think of things that you will just not think of when writing a grant about challenges in how you're that challenges you're likely to meet in conducting your trial. You'll say, oh my God, it's going to be hard for the patient to get from the lab to have their blood draw over to this other place for their, some other test in that period of time. We better put those on different days, those sorts of details. Your grant reviewers will include people who've run into similar problems, and they will know that you have not thought about those details because it doesn't seem like it's really feasible. So when you write your protocol first, you think about a lot more of those details. That allows you to write a much tighter and more convincing summary of the trial procedures in your grant. It also means that you'll have your full trial protocol to include as a grant appendix, and as I've now overemphasized, that can be incredibly valuable. It'll also be there for IRB submissions, regulatory purposes, etc. So where are some pitfalls? So a common pitfall is inconsistency between the grant application and the clinical trial protocol. My impression is that what happens is people spend a lot of time on the grant, then they try to put the protocol together at the last minute, and then they go back and people are reading their grant for them, they revise the grant, they never make the protocol match, or they just didn't have time to really do the crosswalk and make sure they match. That is going to be a marker both of having run out of time and will imply a lack of attention to detail, which is concerning in this setting. And reviewers may or may not take whichever version of the item they think is strongest. There may be inconsistencies in the planned statistical design between the grant application trial protocol and the SAP. Therefore, it's really important, next bullet, to define a hierarchy of documents that says that for statistical analysis, the SAP overrides the protocol, which overrides the grant application. Then once your trial is initiated, so you've gotten the money, you have a protocol, you want, whenever you update any of the documents, for example, you may update the SAP when you realize a data element can't be collected or some other reason. You may update the protocol, even if it's just to add another site. Um, but you want to maintain strict version control. Keep all of your prior versions, change the version numbers whenever you change anything, and then write somewhere, and it can be on a lead page of the updated version, a note, a listing of all the changes that were made from the prior version to the current version. That set of documents over time will create a complete map of the evolution of your trial protocol or SAP from beginning to end, and that is incredibly important when you're trying to get your trial published. So as I mentioned, the statistical analysis plan is a comprehensive and detailed specification of the statistical design, conduct, and stopping rules. And I just want to make it clear that it's the definitive documentation of the statistical design. And there's a reference here, and these slides will be posted, so you don't have to try to grab this now, which is a template for a statistical analysis plan. I don't think it's necessary to follow this particular template, but at least it talks about the elements you want to make sure are included. Many clinical trials that are publicly funded have their documents publicly posted. It's a great idea to look at a set of statistical analysis plan, sort of a smattering of them to see how they're structured and what elements there are to make sure that yours includes all of the relevant elements for your trial. The Manual of Procedures, as I mentioned, is a dynamic document, sometimes called the Manual of Operations. I think that's just so they can call it the moo, but that's just me. That really has the details. It's useful to maintain a log on the front of the mop that summarizes changes. So if you've changed the way you collect an endpoint, initially you thought every patient had to come back for an assessment at 6 months. That turns out to be hard, so you've switched to a phone-based or a video conference-based assessment. You want to have that change, those sorts of changes summarized at the front of the mop. And there's actually on one of the NIH websites, there's a manual of procedures example so you can see what that looks like. Now, I want to talk a little bit about manuscript submission. Why is it so important that you maintain control, version control, and change control of your documents? So ideally, when uh, a publication reporting the results of a clinical trial are submitted, the peer review process should include an evaluation of the original and final clinical trial protocol with a record of the modifications. So the original clinical trial protocol and similarly the original statistical analysis plan are essentially the definition of what was pre-specified. Anything that has changed once you start to enroll patients raise the possibility of bias if those changes were made with knowledge of incoming data. And so you want to be able to explain what you changed and why you changed it. Obviously, adding sites due to, a lower than expected rate of patient enrollment, that's perfectly fine. But changing, for example, a statistical analysis of the primary endpoint should only occur with input from people who are blinded to interim results. So you're not chasing in your analysis some noise in the data that would lead to an increase in the false discovery rate or bias. Peer reviewers may also want to review your DSMB charter, especially if there was an issue with an interim analysis, or even the interim analysis reports if you had early stopping of your trial, for example, for efficacy at an interim analysis. Grant applications do not support this. And I just want to say that one of the things we've seen as a pattern in peer review work is that NIH-supported grants have far inferior documentation at the time of peer review than industry-sponsored research. So in industry, the importance of the hygiene of these documents is well understood, especially if industry is conducting a trial that may create data that they wish to submit to a regulatory agency, whether it's the FDA or the European equivalent. For NIH-supported research, there is, I think, a temptation that once you get the money to just run the trial and not have the end game with respect to submission in mind. And the NIH does not require the level of uh, discipline in this area that regulatory agencies do. So we will routinely, in the grant review, excuse me, in the manuscript review work that I do, we will routinely see NIH-supported work where the manuscript reports an analysis that seems reasonable, but you can't find any documentation that it was pre-specified because it's been 6 years since the grant was prepared. The statistical personnel have turned over, new techniques have become available, lots of stuff has happened, and then it's impossible for the authors to assert that this was a pre-specified analysis plan, which raises all kinds of potential concerns. So in summary, clinical trial documents, including the grant application and the protocol, differ with respect to their purpose, the content, the audience you're trying to influence, and the requirements for revision and retention of audit trails or change documents. If you keep these distinctions in mind, it'll help you draft documents that are effective for their intended purpose and support both the funding, so the application, or the conduct of the clinical trial. So let me stop there. I'm happy to answer questions. Um, if we have time, Megan doesn't know I was going to say this. I'm actually fine if someone wants to put up, for example, a set of specific games and we can talk about it, whatever would be useful for you in the time that we have remaining.
Thanks, Roger. Thank you, everyone. You're welcome to put questions in the chat. You're welcome to raise your hand and unmute and ask Roger. Um, we have plenty of time, so please ask away.
Uh, Hi, this is David Lynn speaking. Can you guys hear me?
Yep, great. Go ahead.
Uh, I really appreciate that presentation. That was terrific. Just a quick question for NIH grants. When you're talking about the clinical trial protocol, is that basically what goes in human subjects, um, in the human subjects section?
So I
Is
think
that sort of
generally,
equivalent, or yeah?
yeah, so the human subjects section, and there's people here who have done a lot more recent NIH reviewing than I have. But the human subject section in my mind is where you discuss the human subjects' concerns or strategies for ensuring that the risk to benefit ratio for human subjects is appropriate. So that's where you talk about consent procedures, inclusion of vulnerable populations, surrogate or proxy or waive consent as appropriate, safety monitoring both at the individual level and at the trial level to ensure the ongoing safety of subjects. So it's really focused on sort of the safety issue. Whereas the detailed clinical trial procedures are going to be in the research plan, data collection activities, those sorts of things. I'm going to pause for a section and make sure that none of the others, Lori, Dr. Silverblight.
I see. So just to clarify, you're saying that the sort of distinction between sort of the science at least for an NIH grant, the distinction in between the science and the clinical trial aspects are both in the grant, but they're just in different sections of the actual research protocol.
So I think there's an issue of the sections, the organization, and how much detail there is. So the NIH grant has to both establish the importance of the underlying question. There's a gap in care, there's a gap in scientific knowledge. So there's a gap, and it is important to fill that gap because it's preventing us from being able to diagnose or treat patients better.
Right, absolutely.
Then the grant has to also convince the grant reviewer that you're going to be able to conduct a clinical trial that has a good chance of closing that gap. And there has to
Sure.
be enough detail in the grant application so that the clinical trial seems, likely to be feasible and rigorous and be able to address the question. There's a That is less detail than the detail about how you're actually going to operationalize the clinical trial, and that's going to be in the protocol, and to some extent in the manual of operations.
Got it. And yeah, that's perfectly clear. The protocol and the manual of operations, those documents at the end of the day are ones that go to local IRB, or those documents are ones that go where exactly in the NIH grant, if at all?
So if you can figure out a way to have your protocol visible to the grant reviewers, you want to do that because there's a lot of detail there, and very often because of page limitations, grant reviewers ask questions about things that the investigator could have answered had they had more space. The art here is how do you put that in front of people? And if your mechanism allows appendices like clinical trial protocols, you want to do that. So it's right there in the application. If it does not, I highly recommend you months ahead create a public site and you make it publicly postable, and you give the URL in your grant. If you have even more time, this is an exception rather than the rule, then what you can do is you can submit and publish a protocol manuscript to a journal like Trials. I have no affiliation with the journal Trials, I should say. Um, but like the journal Trials, which publishes trial protocols, and the details, things like the trial protocol document itself, and even the statistical analysis plan, can be electronic supplements to that protocol publication. What that does is it puts all of that information into the public domain, and now it becomes something that you can reference in your grant, and a grant reviewer who's interested in that level of detail can find. All
Got it.
too
Okay.
often, what happens to people is you're writing a grant at the last minute, you don't have time to do any of this.
Right.
And what I'm trying to emphasize in as gentle a way as I can is that the it will be obvious if you found yourself in that position.
Right. Okay. Perfect. Thank you.
Can
Thanks,
I ask
David.
you a question?
Sure.
Sure.
Hi, I'm Hiro Kinaria from UCLA. So can you comment on the difference between the typical what we think about clinical trial, like FDA regulated, like drug trial, drug versus placebo, or but NIH defines clinical trial more broadly, and we often end up writing a lot about clinical trial related uh, details, protocol based on that. For example, I'm just giving an example. My research is about giving the electrical stimulation from the intracranial electrodes and
Mhm.
observe the behavioral changes such as side effects of the humans or changing the biomarkers.
Mhm.
This doesn't really fit into the typical sense of clinical trial, but NIH thinks this is a clinical trial. How are what are the differences between those two things when it comes to the documents, paperworks, requirements? Do you know
Yeah,
anything about that?
yeah. So I think there's not as much difference as you might expect. And obviously, I don't know the details of your work. So I tend to present these documents as if one is doing a sort of latest stage clinical trial, a phase 2 or phase 3 like trial, where you have a comparison group, you have a primary outcome, that sort of thing. Um, the same rules apply if you're doing more exploratory earlier work, but you just need to be explicit about it. So, for example, if you are um, say you're doing some very early work with an implantable device, and you're going to implant it in 10 people. Just going to make something up, and I hope this
Yeah.
um, isn't crazy. Let's suppose you have people with an intractable pain syndrome, and their life is their quality of life is extraordinarily poor, and you want to test a very innovative implantable device that you think may help manage their pain. And it's never been tried before. And because it's pain, there's not really an effective animal model for it either. So it's highly innovative, but also high risk. You can write a clinical trial protocol for the implantation of the first device followed by a stop and an analysis. And then if it's, deemed safe and there's some evidence of efficacy in that individual, planting it in 2 additional people. So you can do very early exploratory stuff. But for that to be considered a reproducible work, you really do have to have a plan. What is your criteria for success in that first patient? You don't just say, we'll see how they're doing. I think sometimes in early stage work, there's a tendency for there to be a certain amount of vagueness in how success or failure will be assessed and how it will be incorporated into the treatment in the next set of patients. And you want to really think that out in advance. Doesn't mean you can't change it if you see a response completely outside of what you thought was plausible. Okay, but you need to have a plan that's pre-specified. So I think it's not as different as you might think. Now, obviously, if you're doing a single arm trial, there's no description of randomization. Um, I think virtually all of the work associated with this course will require consent or an equivalent procedure. So I think all of those considerations will need to be described. So I'm not sure if that's helpful because I don't know the specifics of what
That's
you're
very
doing.
helpful. That makes sense. I'm thinking more like a setup that is clinically used, but the stimulation, assessing those things, these are not standard of care in any way. So I still need to get the informed consent. I have to describe what I'm going to do, what I'm going to measure. So whatever you said, it makes sense to me.
Okay.
Appreciate it. Thank you.
Well, I'm glad. Okay.
Thanks, Hiroki. We have a couple questions in the chat. The first is, how do reviewers reconcile differences when a strong grant application translates into a weaker or less detailed protocol?
Well, I think it's unpredictable. I think in some funding mechanisms that have extremely competitive pay lines, that is enough to send something back for, to require a revision. You could see in a review a document that says, extremely strong in the significance and relevance, and then there are some concerns about feasibility or reproducibility based on a lack of detail. So I've seen that. I have also seen in some agencies a funding of things in which when we look at the documents when we're doing peer review of the manuscripts, it is absolutely clear that the protocol was inadequately detailed or sometimes frankly absent, and they got federal funding. So I think it's unpredictable. It depends on which study section you're in. It depends on the competitive nature of the process.
Thank you. And then one other question was, do young investigators write and publish protocols in the way you described? This person only seen it done for large RCTs.
Oh, absolutely. Absolutely. So, it is true that the protocols that are published tend to be in larger efforts. I think partially that reflects the experience of the personnel that are involved in those efforts and the long runway those efforts often take in order to get going, so they have time to do that. I think if you have any sort of innovative aspect to your trial design, that publishing a protocol paper makes a lot of sense. I can picture very early stage investigations that for which it probably doesn't make sense, but then it still makes sense. Remember, the purpose of this was to enable that level of detail to be available to the grant reviewers at the time of grant review, in which case, if something really seemed not like a publishable trial design, I would still try to find a way to include it in the application or make it available, say, on a study website. And you can even use your personal website at your university and have a project page and put a protocol there. But the idea is if you've thought about these things in substantial detail, let the reviewer know that you've thought about it in that detail. And then, Rob, do you want to talk about your comment just so people understand that? Dr. Silberlight? He may have had to.
No, I'm just having trouble finding my unmute button.
Yeah.
Which of I did two comments. Which one did you want me to comment on?
I think you should comment on both. I think they're both important.
So clinical trial applications that NIH has been struggling with for years, how to properly structure them into grant applications. They used to allow the protocol to be included as an appendix, but it was overwhelming for reviewers who, had a 12-page grant application to review, but everything they really needed to know was in the 150-page protocol. So they got rid of allowing, for most funding opportunities, allowing the protocol to be included. So they end up having these different sections that came in different packets. And so the reason I think there was some confusion with the human subjects is that there's a bunch of clinical trial stuff that's bundled with the human subjects protection questions in the grant application sections. And so it's like the title says clinical trial human subjects protection, but it's clinical trial and human subjects protection, not. So it gets confusing. That being said, there is a new proposal, trans NIH, to reorganize clinical trial applications into a better form. And there was an RFI that just went out last week, the week before, where they proposed essentially a table of what the new form will be based on. And it's imperfect, but better than what we've been dealing with recently. So if you can find the RFA and look at it and take a look at how they plan to ask for clinical trial information in the future, you can make comments and reply back, and you can get a taste of what the future may look like through this RFI, even if you don't. The other thing that changed, so the clinical trial definition, the very broad clinical trial definition that NIH has used for years was from 2014, and that included pretty much almost all human subjects research was defined as a clinical trial. And some people found that really difficult when it was essentially basic science research using humans. And recently, back in January, they went back and changed the definition to exclude what they call basic experimental studies involving humans. So basic experimental studies involving humans were explicitly included as a clinical trial from 2014 until 2026, and now are not. And the difference is a little subtle to me, but essentially it's when you're not looking at anything, when your study doesn't involve any possibility of affecting the care of patients. So if you're doing something on a human, but not with the intent of fixing a disease, changing a clinical outcome, learning about a treatment, and you're just trying to understand how things work, that's no longer in the NIH definition of clinical trial. It sounded like the description was still going to be clinical trial, was like a single arm clinical trial, the one that you asked about. But that's a change.
Great, thank you. I'm going to, in the last minute or so, try to address the questions in the chat. The comment about a chair letter that would cause me concern. So if I see a chair letter that says, currently Dr. X works in the clinic, 4 days a week, and we will make every effort to provide them with sufficient protected time to work on this. Basically, promises of an effort of a chair. I should be careful. Promises that the chair will try to get you time are always a red flag. They need to say exactly what you're going to have. And the problem is that institutions have restricted chairs and others' ability to write letters like this because the institution says you can't actually promise that we won't have clinical needs that the person will require. So it used to be that chairs could write letters that say someone will have 50% protected time for the 5-year period of the grant. And then they were obligated as an institution to honor that, at least to some extent. So I'm just looking for something that's a lot stronger than we will try. And then sometimes the chair's letters, like the example I gave, point out that the person doesn't have anywhere near enough time to actually do this in the current state. If you start from not having enough protected time, the promise that you'll get more protected time later, I always see as dubious, and I was a chair for 7 years. Yes, you can absolutely write and publish a protocol for a trial that is not funded, for which you never know if you're going to have funding or be able to do it. You're basically drawing a line in the sand of what you intend to do. It's like a detailed clinicaltrials.gov registration. It lets people know what you intend.
Can I add one thing to the chair letter thing while you're reading the rest of them? That
Yep.
is, I think one of the things that I've seen that makes people pause is if you are in a high potential call if they don't mention call, like if you're a neurosurgeon or a neurocritical care doc, and they say, oh, you're only, and they know you don't have a lot of clinic, you know, what your clinical time may be. So sometimes it's helpful to have the chair iterate, you know, kind of say exactly what your call is going to be like, because that's where, like if you're going to be on call every single weekend so that you can have 75% protected time or 50% protected time, that's a problem, right? So some, especially study section, people really think about that for neurocritical care and neurosurgery specifically.
Great. Thank you, Lori. Thank you, everyone. I need to let you get to your next obligation. It's been a pleasure. People can always email me. If you didn't catch my email, it's on the slides, and Megan can give it to you as well. All right. Thank
Roger.
you.
Thank you, everyone.
so
Thank you,
much,
Roger.
Okay.
Be well. Bye.
Bye-bye.